Hi, my name is Samantha Luois. I'm a member of the undergraduate biology research program. My major is molecular and cellular biology, and I'm a junior. So my research is involved with microbubble condensation. Well, a microbubble is a lipid-encoded bubble, essentially, that is filled with a perfluorocarbon. And a perfluorocarbon at the center can either be a gaseous core or a liquid core. Essentially, we inject this microbubble into the bloodstream, and once it's in the bloodstream, we apply ultrasonic energy, and this ultrasonic energy is usually in the form of ultrasound, and once that occurs, we have an illumination of the entire bloodstream. Here, what we do in our specific lab, Terry Matsunaga's lab, and with collaboration with Paul Dayton and Paul Sheeran in our University of North Carolina collaborators, is that we work with this microbubble, and we started out with a gaseous core. And once it's at the gaseous core, we apply super high pressure, just regular ambient room pressure, and then we decrease the temperature in a basic isopropanol water bath. That causes this microbubble to condense down to a liquid core microbubble. And what that does is it essentially gets this bubble small enough to exit the bloodstream. Currently, we work with microbubbles that are used within the vasculature. That means that we can image all kinds of things like the bloodstream, we can image different types of tissue with a ton of angiogenesis going on, but we cannot image anything with the potential to extravasate, so tumors, organs, things without all of that angiogenesis being pumped through it. It's not just good enough to get a bubble that's small enough to extravasate, but also it has to maintain that ability to reflect that ultrasound. Now, once we have that bubble small enough and labile enough to maintain its echogenicity, meaning its ability to reflect that ultrasound, then we can get that bubble to extravasate, exit the bloodstream, and once it has exited the bloodstream, at that point we can apply ultrasound, and then we get the bubble to reflect that ultrasound once it's at its gaseous state once again. And we do that by just applying basic acoustic energy in the form of ultrasound. What we see here is that over time, at let's say an ideal diameter, which tends to be between 0.5 and 2.6 micrometers, the bubble, it maintains its same diameter over 60 minutes. So we have from 0 to 60 minutes, the diameter is constant for that bubble. So that means that the bubble is able to be stable enough for an hour or so. And that means that we can get the bubble to enter the bloodstream, exit the bloodstream, and then we can apply the ultrasound within that 60-minute time period. So essentially, it's extremely ideal in terms of how stable it is. Our lab, the Matsunaga lab, has been exploring different perfluorocarbons at the core. So we have lots of different gases that can be present at the core of the microbubble. So we can have perfluoropentane, which is very unstable, but it maintains a bubble size that's very, very small. And so it's ideal for extravasation, but not for stability. So it's really essentially a trade-off between how stable the bubble is and how small the bubble is. So what we settled on is what's called a gas perfluorobutane. Perfluorobutane has a negative 1.7 degrees Celsius boiling point, and that's ideal for stability in the vasculature. So once we get the bubble to extravasate, it's important that this bubble can now be used to image leaky vasculature, can be used to image all kinds of things that don't have angiogenesis being pumped in through them. And that has potential for imaging tissues, the liver, different organs, because the bubble now is small enough.